Good morning, everyone, and good evening to some of you.
Right. Good morning, everyone,
And good evening to some of you. Welcome to, I hope everyone can hear me
welcome to,
well.
well, I hope everyone can hear me well.
I can hear you pretty well.
I can hear you
I can
pretty
hear
well.
myself twice. So I
I
think
can hear
that's the
myself
total
twice.
part. So,
That's
okay, so my name
okay.
is Zam, and today
Yeah,
I'll
so
be
my
your
name is
host.
Zam, and
It's a
today
real
I'll be your
privilege
host.
to welcome you. It's a
It's a
real
real
privilege.
privilege to
AI
welcome
for
you
Life
to
was created as a community for life science
AI4Life
researchers to share ideas, knowledge, and
and
experience around cutting-edge AI technologies. The
AI
Innovation Talk
technologies.
series are
The
designed to be
innovation
a
talks
short,
series
focused session where tool builders, developer like yourself, and also
like
researcher
yourself
explain
and
how
also
their
researcher
platform and
explain
model
how
works, where they can be useful, what limitation and matters, and how it can fit into real research workflows.
the
You can think of
workflows
it as a practical forum to explore
to
tools that can help drive
explore
your
tools that
workflows.
can help drive your
So today
workflows.
is my
So
great honors to introduce our speaker, Dr. Ying-Ja Chen. Dr. Chen is a solution
Dr. Chen
architect
is
for
a
healthcare
solution
and life
for
science at
healthcare
NVIDIA.
and life sciences
She
at NVIDIA.
specializes in AI for digital
Specializes in
health,
AI for
genomics,
digital health,
drug discovery,
drug discovery,
diagnostics, and medical
diagnostics,
devices.
and
And that is going to be
and
the
that
focus of
will be
today's
the
session.
focus of today's
And I
session.
hope it will spark
And I hope it
ideas
will spark
relevant to your own
ideas
research and
relevant to your
pipeline.
own research and
Our
pipeline.
agenda today
Our
include a
agenda today
presentation from
includes
Dr.
a
Chen
presentation from
on
Dr. Chen
NVIDIA
on
BioNeMo.
NVIDIA
I'm sure
BioNeMo.
some of you have heard
I'm
of it,
sure some of you have heard it.
covering the framing
Covering
of
the
generative AI in
framing
life
of
science
generative
and
AI in life
discovery,
science and discovery,
a foundational
a
introduction to
foundational
the
introduction
platform's
to
core
the platform's
pillars,
core pillars
and practical
and practical case studies
case studies showing BioNeMo in
showing how
action.
in action.
We'll then move into
We'll then
Q&A
move into
session
Q&A
with
session with
questions submitted by the
questions submitted
audience
by the
if
audience
time
if
allows,
time allows,
a live
a
discussion.
live discussion.
With that,
With that,
I'm excited
I'm
to hand the floor
excited to
over to
have
Ying-Ja
and
to
for
take us to
India
the frontier of
to take
generative AI
us
for
to the
life
frontier
science.
of AI for life science.
Over to you. Over to you.
Hi, thank you,
Hi,
Zhang,
thank
for
you, Jan,
the
for
introduction. I think you guys should be able to see my slides in the cover page right now. Today, I'm very excited to show you the latest from NVIDIA, and the product name is called BioNeMo, but this is basically our generative AI platform for the computational biology and drug discovery space. When we look at how biology and AI come together, in the beginning, you really need biology discoveries are still, even today, are still driven by experiments. Experiments are the fundamentals and the state of the art of how discoveries are made. But nowadays, um, we begin to build foundation models and have model-driven discovery. These models seems to understand certain data types within our space, like our macromolecules, like our DNA, RNA, and proteins. And because they look at these as language, and they are like language models that understand certain type of data. And because of that, they are able to find patterns and insights so we can make discoveries based on these models. And in the next wave, we believe that we will go through a closed loop discovery where we have models, AI working with experiments in a lab in a loop manner. And let me show you how that works, or this is still more conceptual. But nowadays, you can see in the wet lab, you have all of your in vivo, in vitro experiments generating a lot of high throughput experiments, generating lots of data. And these data can go through, that you can now send that to the dry lab, or what we call an AI factory, and learn these biological insights. And after the models learn, it's actually able to design new molecules and design new or determine new hypotheses so that we can make molecules and go back to the wet lab for testing and iterate through so that we get more and more intelligent models and make new discoveries. At NVIDIA, our BioNeMo platform, people used to think of it as models or frameworks that help accelerate these models, but now it's really taking you through the whole AI lifecycle for biological generative AI. So we have foundation models, but we also, more importantly, for these models, you need to have data. And we also start working on providing open data sets. So we believe in open source and open science. So most of our tools are open, and we also release the most open models in the world. So within BioNeMo, some of the open data sets include like contributing to the AlphaFold protein complex data sets. So we have these physics-based or data, different kind of models and data sets. And within our models, we go from understanding biology, like the foundation models, or going into designing and like generation models, and also have molecular property prediction models that are very important. Besides these models, we provide libraries. These libraries are things that help you train models, customize models, evaluate models, or some of them are for manipulating data, because a lot of times the computational biologist is actually spending time um, converting data from one format to another format using various tools, and we accelerate these tools and provide libraries. We also have training and customizing recipes. I will touch upon that today. And at last, many of you probably don't care about training models, but you care about using models. So in deployment, we optimize these models for the best performance. They run faster with the highest throughput and with the best inference speed so that you can that will be ready to use for a researcher. And in that part, we have our technology that can accelerate and optimize these models. For deployment, we also provide blueprints, which are workflows that put these models together into applications so you can understand and don't get too lost in the vast wealth of models. So let's look into a little more detail. These are the names of each of these tools within BioNeMo. So there are lots of them, and I will try to cover all of them today, including data sets I've mentioned. We contribute to the AlphaFold protein complex structure database. In terms of models, we developed many, NVIDIA's research team trained many foundation models covering DNA, RNA, protein, and small molecule space. We provide libraries and tools to work with these. And training, we have training recipes for deployment. I will go through a few blueprints today. So first, let me introduce you to NIM. NIM stands for NVIDIA Inference Microservices. So by the name, you can tell it is a microservice. So it is containerized in a Docker container, and that allows easy install and also calling API, so easy to use through an API. But most important of all, every model that we drop into in NIM, we actually optimize for the lower layer parallel processing part of the GPU so that in a lot of times a NIM model will run 2 to 5 times faster or higher throughput than the same open source model. And I'll show you in the next slide what are the NIMs. You can think of these as individual models, and these are the NIMs that are available within BioNeMo for biology. Uh, on the top left, we see OpenFold 2, OpenFold 3, and BOATS 2. These are all protein structure prediction models. And the OpenFold project is actually a community-led open source project to rewrite AlphaFold. And so OpenFold 3 benchmarks against AlphaFold 3. It also offers protein and small molecule or protein-protein, protein nucleic acid complex structure predictions. Same for BOATS 2 that was developed by MIT, and we made them into NIMS more optimized. In the middle, we see DiffDock, standing for diffusion docking. It is an AI-based molecular docking tool that is much faster than traditional molecular docking tools and excel in blind docking. DiffDock, I think it's already at 3.0 now, at the latest. And then on the right-hand side, RF diffusion and protein MPNN is the David Baker Nobel Nobel Prize winning model that does protein design. So based on a protein of interest, if you want to design a target protein, RF diffusion will do that, and protein MPNN will help you assign the amino acid sequence within that designed protein. On the bottom, we see MoMeM and GenMo. These are small molecule foundation models, generative models that are developed by NVIDIA. And so these are small molecule generators that can generate drug-like compounds or compounds and that you can design your oracle function, like what you want to generate towards. And a lot of these AI-based generation generates molecules that medicinal chemists never thought about. But so later on, we have other tools that help you design the reaction synthesis path. In the middle, EVO2 is a DNA foundation model trained on reference genomes across all species from prokaryotes, archaea to eukaryotes. And Atlas MSA search is a multiple sequence alignment search. This is a step that is important for protein structure predictors such as OpenFold that is actually used as part of these structural predictors. But this MSA search is actually the bottleneck step that makes protein structure prediction run the longest. So we also provide a separate NIM that accelerate this part. So what can you do with these models? Let me show you two blueprints, which are applications for these models. The first one is called generative virtual screening. Here, traditionally, when a pharmaceutical company want to do drug screening, they have millions or billions of compounds in 96 or many well plates, and they have robotic arms coming in with an assay that will screen through each of these compounds. So it's a major investment. But here, what we can do is we can do things virtually. First, if you have a target protein of interest and that doesn't actually have a crystal structure yet, you can use the top part using OpenFold 2, which needs to go through MSA search and a protein database to get the multiple sequence alignment and finally predict the protein structure of your target protein. On the bottom part, you see we can use GenMo, this model, to generate a bunch of small molecule libraries, and this can be a fragment-based method. You can start from a scaffold or start from nothing and generate a lot of small molecules. And then on the right-hand side, we will dock each of these small molecules to the target protein using diffdock for a fast screening. And finally, you can screen through a large library of compounds and arrive at a smaller subset that has the potential to bind to your target protein. And so after that, of course, you still need to do experimental validation, but you can um, imagine this type of virtual screening is saves a lot of time and money for the people doing drug screening. Let me show you a second blueprint. This one is called Generative Protein Binder Design. Now, another way, other than screening for small molecules that bind to your target protein, another way to find drugs is to design a small protein against your target protein. So the top part is the same as the previous slide. We predict the protein structure of your target protein if it's not already available. But on the bottom is the main thing. First, RF diffusion is a model that can design a protein fold, like a 3D structure that binds to your target protein. You can set where is the interface, where are some hotspots, and a target size of this new generated protein. And after it designs like the fold, then the next step is using protein MPNN to find the amino acid sequence that can fit into that will fold into that protein structure. You can generate many sequences that fold roughly into the same structure. So at last, we use AlphaFold to multimer to predict the protein complex structure. That is the designed protein binder with your target protein, and look at the structure in complex as a validation step to make sure what you design actually binds to the target protein. Besides the NIM models, we also offer developed Clara Open models. These are open sourced models available on GitHub. And these are foundation models trained by our digital biology lab. So this is not a complete list, but very close, and these are probably the ones of most interest. So first, La Proteina and Proteina complexa is a model for protein generation. So in the previous slide, I showed you a blueprint for doing a two-step generation of a protein binder. Using La Proteina or Proteina complexa, you can actually do generation of the protein fold and the sequence in one step. And it scales up to 800 amino acids, which is pretty impressive using this model. The next codon FM, by the name you can tell, is an mRNA foundation model that is codon aware. And I'll show you one application in the next slide. We also have RNA-Pro for RNA protein structure prediction. The next one, Reason, by the name, it's a reaction synthesis pathway predictor for small molecules. And Kermit is a small molecule molecular property prediction model or tool. So let me show you an example of how to use foundation models. So here is an example on Codon FM. And the goal here is to predict synonymous variant prediction. So to tell if a synonymous variant is actually pathogenic or benign. Usually we think of them as benign, but for variant prediction, it's actually a hard problem. And here, because the codon FM was, it's a family of foundation models. So most of these models name is actually in codon with the parameter size. There are different sizes that come with it. And these models are used to generate embeddings on the mRNA sequences. So this was used to look at all of the pathogenic versus benign variants in the ClinVar database. And after 50 iterations of, um, of training, matching, it has to be gene position and gene level normalized. And this model was able to identify and predict the patho uh, the predict whether a variant is pathogenic or benign. And as shown in the graph on the right-hand side, on the top three, the three best performance are all the codon foundation models. BioNeMo also offers training capabilities. So in the previous few slides, I show you inference, how to use, like what are these models used for? But what if the model is not good enough? Because, for example, we can start from protein foundation models that understand protein language, but then maybe you are working on a specific enzyme and you want to tailor to enzyme type of models, and you want to customize and train, um, further train the model or do fine tuning. BioNeMo framework has what we call BioNeMo recipes that are many steps that help you optimize your model. And we also have tools for doing customization and evaluation. So the BioNeMo framework really takes you through the whole life cycle from data loading to um, to training and customization, making predictions. Inference is an important step in training, evaluating the model with various metrics, and after you iterate through model fine-tuning, finally for deployment. You can do all of that following recipes in BioNeMo framework. Oh yeah, so here I'm showing you the advantage of using BioNeMo recipes. It's flexible in scaling. You can start from many um, pre-trained models or frameworks, and the most important is still the throughput that you can get. So on the right-hand side, I'm showing you tokens per second um, of an ESM2 model, with the gray being the baseline model from open source ESM2, and with after a TE, the transformer engine optimization, you get better throughput. If you add an additional sequence packing, you can get even better throughput in terms of training the ESM2 model. So the value proposition for using BioNeMo framework includes that it's domain-specific workflows. We have pre-trained models. These help you seamlessly scale to multiple GPUs with large protein sizes. We really very quickly need to use multiple GPUs, and we provide full stack optimization. So let me show you one real use case of BioNeMo. This is from Dino Therapeutics. It's a startup company that specializes in gene therapy, and um, they want to build a model that can help them um, redesign the capsid protein of an AAV vector, because AAV adeno-associated virus are vectors that are the best for delivering gene therapies known so far. And but they don't deliver to every tissue you want. And they wanted to deliver to neuronal cells or cells in the eye, so they built on top of their own in vivo data sets by using BioNeMo framework to train their protein language model and design and um, optimize or mutate and change their capsid proteins so that it will deliver precisely to the neuronal cells. And you can see um, that the GFP shows how they were able to really deliver to these cells that were predicted by their model. And so now let me show you one slide on how BioNeMo works on the Vecura platform. This is an example using the Proteina complexa, that's the protein binder generator. And here the question was that the researcher wanted to design a target protein that can bind to a binder protein that can bind to the NEK2 protein. And NEK2 is part of the is a protein that's related to DNA replication and will be related to cancer. So if you can block NEK2, then potentially you can block cancer. And we also know that it has an ATP binding site. And so presumably we want our protein to bind at the binding site so you can block ATP from coming in. And so on the slide here, you can see this is a screenshot of what the Vecura platform looks like. Um, with this platform, it's nice that you won't need to know how to write the command for each of these models, and everything is already in in a nice user interface. And so you can choose Proteina complexa, and then you choose the target protein, the PDB file here, and set up basic parameters such as specifying where the hotspot is for the ATP binding site. And within less than 3 minutes, it generates um, a new protein binder as shown in orange here. And it visualizes on the right hand side of the screen, but I also pull out a screenshot to show how this protein that was designed fits into um, the ATP binding sites. And you can look into that all on the Vecura platform. So in the last part, I want to show you a new thing called BioNeMo Agent Toolkit. And this is, I think this is very exciting that now we're entering an AI agent era. And it really removes the entry barrier for a lot of research scientists who don't really know how to write programs, and it would be too cumbersome to learn programming. Now you don't need to learn programming anymore because we have agents. Here is the evolution of AI agents. Not very long ago, 2022, we had ChatGPT, and that changed the world. Everybody is using these large language models nowadays. And 3 years later, not even 3 years, early of 2025, the DeepSeek model came out, and it was revolutionary. It's a much smaller model, but it has these capabilities called reasoning, where you keep generating inference and then let the model think inference through, then it becomes smarter and can answer better questions. And it's only been a year. Earlier this year, OpenClaw came out. And the beauty of OpenClaw is that in the past, if with ChatGPT and DeepSeek, you ask a question and the model will answer. So if I ask it to run protein binder design, it will tell you that you can use Proteina complexa, you can set parameters, here is the code, and you still need to run it yourself. But OpenClaw will do it for you. That's the beauty of the modern day AI agents. And OpenClaw will improve itself, it'll learn your behavior, your style, and then adhere to your style. And nowadays, it's only been a few months, but nowadays everybody's using coding agents such as Cloud Code or Codex, or so-called the ChatGPT app, which does what I just talked about. It not only answer your question, it actually do things for you. But in order for these to do things, so how does it integrate with BioNeMo? Oh, before we look into that, I just wanted to point out that in a KPMG survey, it shows that in the life sciences or biopharma industry, people using agents are beginning to pick up. Like 48% of researchers are already using agents for doing biomarker discovery or drug discovery. And so let me show you how these agents work. This is NVIDIA's NemoClaw blueprint, and it's the same idea that your user will use prompting to ask the agent to do some work, and um, and what the OpenClaw or Cloud Code or Codex, those are what we call harnesses in the middle. The um, it doesn't matter which LLM you use, but this harness allows this LLM to first analyze your question and then and and sort out like what you want to do and then make a plan and finally decide on some of the actions that it needs to take. It probably writes a small script and run that script itself to take action, and it will do the work for you. In order to do the right work, then it needs tools and skills. Tools are just what I just introduced earlier in this talk. All of the models are the tools that are available to the agent. What are skills? Skills is actually a file that writes in natural language what are the instructions to doing something. For example, a skill for running the Proteina complexa model is will probably say what you need to find inputs, protein sequences, protein structure files, and you need to set parameters, and then here is the command. And so with these instructions, the agent will be able to understand and run the right command to do things for you. So we now provide BioNeMo Agent Toolkit that that is a bunch of skills that teaches your AI agent how to run each of the models. And that is really great. So you won't need to learn any of the detailed commands anymore. You don't have to worry about typing a typo or misspelling anything because your agent will take care of that. And your agent will have the right skills to run everything. We worry about the agents because these are autonomous agents running things and doing a lot of things, we worry about security issue. And that is why we put this all in a open shell sandbox you see on the bottom. And this open shell will control the security, what kind of data flow or permissions and authentication can be granted to the agent. And I believe agentic workflow is also being integrated into the Vecura platform. And so besides, within these agents, you can use any of the LLM models, but let me introduce you to the NVIDIA's open model called Nemotron 3 family of open models. We released these this year, and they come in three sizes. The Nano is a smaller 30B with only 3 billion parameters activated each time. And the Super is the more general use 120B model. And we recently also released the Ultra, that is the largest and most intelligent models at 550 billion parameters. So these are open models that are at NVIDIA, we actually release and open up our best models and provide to the community. And I know that the NYB team is also integrating the Nemotron 3 Ultra so that you can get the smartest models and help you integrate into your agents. And so you only need to do prompting, and the agent will run all of these research discovery work for you. Let me show you at the end one example of putting all of these together in an agentic way. So here, we want the agent to do some research on cystic fibrosis and design a drug. So the agent needs to do some planning. It starts from thinking that, oh, it needs to read some papers, so it does a PubMed search. It reads through the abstract, and after reading through these abstracts or these papers, then it has to come up with a hypothesis and identify the target protein for cystic fibrosis. After identifying that, it calls a tool to help design a drug. The tool it called is the virtual drug screening blueprint that I introduced earlier. So with this tool, it pulls out uh, the PDB structure of the target protein. It generates a bunch of small molecules and dock them one by one to the target protein and select by the score. At last, the agent writes a report and generates this report with all of the scoring information to explain to the user. We can ask it to iterate through and critique its own work before it generates the final report to us. So I think with this example, you can see how nowadays you really don't need to know how to write code, but you need to learn about what are the different models or tools that are available there to do any kind of discovery that you would like to make. So at last, let me summarize what we talked about today. I introduced you to the BioNeMo platform, which includes many models or blueprints that put together these models into applications and allow you to either make predictions or generate new molecules or analyze and molecular properties. And finally, I also talked about how we can use agents to do prompting and let these agents run models for you and do discoveries for you. And with that, I'm happy to take questions. Thank you.
Awesome. Thank you, Ying-Ja, for that super insightful presentation and showing us, the practical application of BioNeMo. So we will now move into the Q&A portion of today's sessions. So before this meeting, we have received several questions from participants, our participants here. So I'm just gonna selectively pick a few here. So question number one, how useful is BioNeMo to perform tasks with understudy and novel proteins? For example, plant gene families.
Oh, um, great question. So, um, I think BioNeMo is actually very useful in helping you to understand these novel proteins or understudy proteins. There are limitations, like a lot of these models can't work with very large sequence and proteins, but you can analyze sequences and break down into domains. And because some of the models, like for example, you can take a protein structure prediction model and then you can generate mutations and then look at how the structure changes, right? And so that is one way to identify potential like different variants or potentials, um, well, not really functional, but structural analysis of proteins. Or you can use DNA or like the RNA, like the codon foundation model, to look at variant effects in your models. So there are various ways you can think about how to study them and then help you use these in silico methods to do analysis first before you need to jump to an actual experiment. And also, you can also design new proteins. I don't know about like if you're trying to study a protein itself, but maybe you can generate something similar and or do similarity analysis from embeddings that were generated from these models. Embeddings are just a machine readable language, but you can take your protein sequence and then generate embeddings with like an ESN model or some of these pre-trained amino acid models, and then from the embeddings, maybe you want to make different kind of predictions or generate different properties. It'll allow you to do that. So this is a new way of doing discovery.
Thank you for that. So I'm sure some of us, including me, are particularly interested in plants. We'll definitely take your advice into our flow. So the second question that we have here today is, will BioNeMo be able to gather data sets of different formats, normalize them, and then analyze? How accurately does it handle data sets and pre-process them accordingly?
So what we discussed today, like BioNeMo, is a collection of foundation models and tools. Um, it's I think what you want is, it sounds like what you want is really what an agent's doing. You want an agent to help you process your data, normalize it, and do analysis. And it's great that we have BioNeMo agent toolkits and these agents nowadays, because um, I don't think you will be able to let BioNeMo automatically do that, but your agent can help you. Um, Within BioNeMo Agent Toolkit, Parabricks, our genomic analysis tool, is also available, meaning that your toolkit has skills to run it. And so you will probably need your agent to have the skill to run the right tools, the tools that know what kind of data set you're manipulating, and then do the normalization and these steps using your agent. That would be my suggestion.
Okay. So we have another very focused and particular questions. As a biotechnology student who recently used VQE quantum simulation for molecular energy estimation in nitrogen fixation, I'm curious how NVIDIA BioNeMo handle molecular property prediction for catalyst discovery. Can generative AI models like BioNeMo complement quantum chemistry workflows? for accelerating sustainable biotech research?
Yes, so BioNeMo, I think VQE or these kind of quantum chemistry methods have their strengths. And BioNeMo or these generative AI methods are definitely not here to replace them, but they can work together. So the beauty of Gen AI is that you go from data and you can quickly get you can generate a lot of candidates and you can quickly get a bunch of possible molecules. And and because you with with like quantum chemistry, you can go into more details, or like for force-based methods, you can go to more deterministic details of each of these molecular design. So I would do a two-step process. I will start with a more Gen AI method using some of the models in BioNeMo to do um, molecular property generation or prediction, and then maybe generate a bunch of candidates that you want to study, screen for them using whatever Gen AI tools you already have, and then take the most promising ones and then do a more quantum chemistry based method and refine on your results.
Thank you, Ying-Ja. So I believe that we have the floor for perhaps one uh, live question. So if anyone is curious or have any questions, please feel free to open your mic and share. Okay, so thank you, Ying-Ja, for addressing these questions and sharing your perspective. As we wrap up, I want to sincerely thank uh, Dr. Ying-Ja Chen for sharing her expertise and walking us through the BioNeMo platform. Super useful. And thank you all for joining and engaging in today's discussions. This webinar is part of AI4Life Innovation Talks series, where we spotlight frontier AI tools that can help drive research forward. We encourage you to explore model discussed today. Check out our platform at Vecura for streamlined molecular modeling. Continue the conversation within the AI for Life community. We look forward to seeing you at our next AI for Life Innovation Talks session. Until then, thank you again for your time and participation. Have a wonderful day. Thank you, Ying-Ja.
Thank you.
Thank you.